بسم الله الرحمن الرحيم. This is my second video about thymic stromal lymphopoietin in dermatologic diseases, and I'm reminding you again to subscribe to the channel and activating the alarming bell if you are interested in getting any new video. So as I said in the first video, that the role of thymic stromal lymphopoietin in psoriasis could be through increasing the production of interleukin-23 from dendrocytes, which will in turn increase the production of interleukin-17. And I found this nice paper talking about this relation. This paper was published in Clinical Dermatology Review in 2020, December 2020, and it's titled Thymic Stromal Lymphopoietin in Dermatologic Diseases. So in the mentioned paper, they said that thymic stromal lymphopoietin is a cytokine that can act on dendritic cells via two pathways. The first pathway is through toll-like receptors, which will result in a decrease in interleukin 12, interleukin 23 P40. We know that P40 is a common subunit for interleukin 23 and interleukin 12. And so when they stimulate the dendritic cells through toll-like receptors, will decrease this subunit, leading to predominance of T helper 2 cytokines, as the situation in atopic patients. The second pathway is through CD40 ligand, which leads to an increase in interleukin 12, interleukin 23 P40, and subsequently this will increase interleukin 23, resulting in predominance of T helper 17 cytokines as observed in psoriasis patients. This diagram can make things more clear, that we have here thymic stromal lymphopoietin, which is secreted from keratinocytes. This thymic stromal lymphopoietin will act in either two pathways on dendritic cells. The first pathway through toll-like receptors, which will result in decreasing interleukin 12, interleukin 23 P40. And when this one is decreased, there will be activation of T helper 2 inflammation through acting on innate lymphoid cells type 2, T helper cells type 2 cells, and this will activate the atopic dermatitis. While the other pathway is through activation of CD40L ligand on dendrocytes, which will result in increasing interleukin 12 to interleukin 23 P40, and this in turn will increase interleukin 23, which will act on T helper 17 to make T helper 17 inflammation, which will lead to psoriasis. The genetic background of the patient or the type of thymic stromal lymphopoietin can determine which pathway thymic stromal lymphopoietin will follow. Actually, the long isoform of thymic stromal lymphopoietin is biased towards CD40 ligand, resulting in increase in interleukin 23 and development of psoriasis. Now I'm going to talk about the role of thymic stromal lymphopoietin in another cutaneous disease, which is Bullous Pemphigoid. And again, I'm reminding you by my first reference, which is Thymic stromal lymphopoietin in cutaneous immune-mediated diseases, published in Frontiers in Immunology 2021. Although the exact pathophysiology of Bullous Pemphigoid remains uncertain, it's known that autoantibodies targeting structured proteins in the dermo-epidermal junctions and T helper 2 immunity play a role in this condition. Subepidermal blister formation in Bullous Pemphigoid involves autoantibodies directed against hemidesmosomes proteins, Bullous Pemphigoid 180 and Bullous Pemphigoid 230, leading to complement activation, muscle degranulation, neutrophilic and eosinophilic infiltration, and proteolysis at the dermo-epidermal junction. Also, type 2 inflammation includes T helper 2 CD4 T cells and the group 2 innate lymphoid cells, which will produce type 2 cytokines interleukin 4, 5, and 13, and B cells that secrete IgE, and the granulocytes such as eosinophils, mast cells, basophils. And we know that thymic stromal lymphopoietin is a main and important mediator of type 2 immune responses. Actually, several research studies have found significant elevated levels of thymic stromal lymphopoietin in skin lesions and also in blister fluid and serum of bullous pemphigoid patients, particularly in the lower epidermal layer. A correlation was observed between thymic stromal lymphopoietin levels in the serum and those in the blisters, suggesting that thymic stromal lymphopoietin plays a role in the immunopathogenesis of bullous pemphigoid and may be secreted by epidermal keratinocytes in bullous pemphigoid patients. However, the mechanism by which dysfunction of Bullous pemphigoid 180 in keratinocytes leads to the release of thymic stromal lymphopoietin and the molecular pathways and interactions involved in this process require further investigation and clarification. Another issue in Bullous pemphigoid patients is persistent pruritus. Persistent pruritus is the most troublesome symptom experienced by Bullous pemphigoid patients, which has significant impact on their quality of life. And as the thymic stromal lymphopoietin is known to play an important role in inducing itching in various pruritic skin diseases, thymic stromal lymphopoietin could also contribute to the itching experienced by bullous pemphigoid patients. Now I'm going to talk about the role of thymic stromal lymphopoietin in systemic sclerosis. Systemic sclerosis is an autoimmune disorder that is associated with significant morbidity and mortality and is characterized by fibrosis of the skin and internal organs, as well as vasculopathy. The three key or main features of systemic sclerosis are vascular dysfunction, autoimmunity, particularly the involvement of T helper 2 and T helper 17 responses, and fibrosis. And thymic stromal lymphopoietin was found to play an important role in the whole process, starting from the pro-inflammatory and pro-fibrotic stage of systemic sclerosis. Several studies have shown that the expression of thymic stromal lymphopoietin is markedly increased in the skin of systemic sclerosis, like mouse model and patients. However, the factors contributing to the production and the precise mechanisms that initiate the expression of thymic stromal lymphopoietin in systemic sclerosis are not understood. Previous studies have shown that toll-like receptors and interleukin-13, interferons, and tumor necrosis factor superfamily member light can stimulate the release of thymic stromal lymphopoietin in systemic sclerosis. There is also an elevated quantity of endothelial cells that produce thymic stromal lymphopoietin in the dermis, which was strongly correlated with skin fibrosis in systemic sclerosis. Also, serum level of thymic stromal lymphopoietin were increased in systemic sclerosis patients and linked to vascular dysfunction, which suggests that thymic stromal lymphopoietin may be involved earlier in the progression of systemic sclerosis, as vasculopathy occurs at an early stage of the disease. In this diagram, we can see the proposed role of thymic stromal lymphopoietin in systemic sclerosis. We have here thymic stromal lymphopoietin, which comes from keratinocytes or from fibroblasts, and also from the endothelial cells through activation by interleukin 1 beta and 13 released from the platelets. And this thymic stromal lymphopoietin will lead early to activation of both T helper 2 inflammation and T helper 9, which is sharing in the autoimmune process. And lately, this thymic stromal lymphopoietin with help or interaction with transforming growth factor beta will work on fibroblasts to induce fibrosis. The production of thymic stromal lymphopoietin by human endothelial cells is induced in a dose-dependent manner by platelet-derived interleukin 1 beta, which can contribute to skin fibrosis. Also, light, which is tumor necrosis factor superfamily member 14, also can stimulate the production of thymic stromal lymphopoietin in keratinocytes and work together with transforming growth factor beta to further impact the thymic stromal lymphopoietin expression. The research findings indicate that a complex interplay between transforming growth factor beta and thymic stromal lymphopoietin, and also highlight the essential role of thymic stromal lymphopoietin in transforming growth factor beta mediated skin fibrosis. So a comprehensive understanding of the relationship between the main cytokines, which includes transforming growth factor beta, interleukin 13, and thymic stromal lymphopoietin, is critical for the development of effective treatments for systemic sclerosis. Research has shown that a significant increase in thymic stromal lymphopoietin and interleukin 4 levels in systemic sclerosis patients, which are closely linked to the elevation of T helper 9 responses. T helper 9 responses play a role in the development of autoimmunity and the production of autoantibodies in systemic sclerosis. We can conclude that thymic stromal lymphopoietin has a significant involvement in both early stages endothelial injury and autoimmune inflammation, and later stages fibrosis of systemic sclerosis. So, targeted modulation of thymic stromal lymphopoietin could be a promising new antifibrotic strategy in systemic sclerosis patients. As regards the role of thymic stromal lymphopoietin in chronic spontaneous urticaria, in chronic spontaneous urticaria, mast cells are affected by interleukin 25, 33, and thymic stromal lymphopoietin. And they have already found that a significant increase in thymic stromal lymphopoietin positive cells in the affected skin of patients with chronic spontaneous urticaria as compared to non-lesional skin and healthy controls. And these findings suggest that the involvement of thymic stromal lymphopoietin in the development of chronic spontaneous urticaria and indicate that the blocking of thymic stromal lymphopoietin receptor signaling could be a potential effective treatment for chronic spontaneous urticaria. And now I'm going to talk about another disease, which is Behçet's disease, and the role of thymic stromal lymphopoietin in it. Actually, the messenger RNA expression level of thymic stromal lymphopoietin and interleukin 33 were significantly elevated in the skin lesion of Behçet disease patients and were positively correlated with the disease activity, indicating the important role of the thymic stromal lymphopoietin and interleukin 33 axis in the pathogenesis of Behçet disease. So this pathway may link environmental triggers to systemic immune disturbances in Behçet disease. As regards vitiligo, the main explanation for the development of vitiligo is the autoinflammatory theory, which is frequently linked to other autoimmune conditions such as thyroiditis. Genetic studies have identified certain variants of thymic stromal lymphopoietin that may serve as crucial genetic predispositions in vitiligo, with these variants being associated with a higher susceptibility or incidence of vitiligo. Recently, there was a case control study that was carried out to clarify the connection between thymic stromal lymphopoietin messenger RNA expression and vitiligo. The finding confirmed that patients with generalized non-segmental vitiligo had significantly lower thymic stromal lymphopoietin messenger RNA expression compared to healthy controls. However, further research is required to determine the expression of thymic stromal lymphopoietin at the protein levels in the serum and the lesional skin of vitiligo patients. So this study was done on the thymic stromal lymphopoietin messenger RNA, not the protein thymic stromal lymphopoietin. Now I am going to talk about the role of thymic stromal lymphopoietin in rosacea. And although rosacea occurs in sebaceous gland-rich skin, affected skin of patients with rosacea is frequently observed as severely dry and very sensitive skin. In healthy donors with sebaceous rich skin and no rosacea, thymic stromal lymphopoietin expression was found to be elevated although the dendritic cells were observed to be in an immature state. This indicates a possible tolerogenic role of thymic stromal lymphopoietin in skin areas with abundant or rich sebaceous glands. However, in papulopustular rosacea, a breakdown of this immune tolerance is evident, as evidenced by the activation of skin dendritic cells and changes in the inflammatory profile, leading to skin barrier dysfunction and the predominance of T helper 1, T helper 17 immune response. This paper was published in Journal of Investigative Dermatology in 2017. Its title is Sebaceous Gland Rich Skin is Characterized by Thymic Stromal Lymphopoietin Expression and Distinct Immune Surveillance, which is disturbed in rosacea. According to the results of the previous paper, they hypothesized that altered sebum composition could be responsible for the loss of tolerogenic thymic stromal lymphopoietin, which may be one of the first steps in the disruption of the non-inflammatory state. So in this diagram, we can see that in sebaceous gland rich skin, which is normal or healthy, we have normal level of thymic stromal lymphopoietin, which make the dendrocyte in a low activation state or immature state. So we have in a state of non-inflammatory, where this dendrocytes stimulate T helper 17 beta and Treg. We will talk about T helper 17 beta, which is low active or is non-inflammatory like the regular one, and they have also Treg. And this T helper 17 beta will produce interleukin 10 beside interleukin 17. While in cases of papulopustular rosacea skin, which is also rich in sebaceous glands, we have a lower level of thymic stromal lymphopoietin due to changes in the sebaceous gland rich specific microbiome and sebum content. So we lose this tolerogenic effect of thymic stromal lymphopoietin on dendrocytes, and we have the more activation of dendrocytes, which will secrete or result in activation of T helper 1723, the very inflammatory one, and T helper 1. So we have too much amount of interleukin 17 and interferon gamma as an activation of T helper 17 and T helper 1. And now we have this inflammatory situation in papulopustular rosacea. In the presence of thymic stromal lymphopoietin produced by keratinocytes in sebaceous gland rich skin, which is normal, infiltrating dendrocytes are in a non-inflammatory stage, and T cells are Tregs and T helper 17 beta cells, while in papulopustular rosacea, altered microbiome and sebum composition will lead to decreased thymic stromal lymphopoietin production, and many inflammatory cells will appear like dendrocytes, T cells, macrophages, mast cells, and neutrophils, and the disruption will occur of the previous non-inflammatory situation. In papulopustular rosacea, an interleukin 17 interferon gamma type inflammation will happen, which means that we have T helper 17, 23, the other type, and T helper 1 inflammation here. And because interleukin 17 is able to further inhibit thymic stromal lymphopoietin production, so this will lead to a vicious circle. In this scenario, T helper 17 cells were divided into two types: non-pathogenic T helper 17 beta and a pathogenic T helper 17 23. T helper 17 beta cells are characterized by the production of interleukin 17 with interleukin 10 and the expression of retinoic orphan receptor gamma T, while the more inflammatory one, T helper 17 23 cells, which play an important role in the development of inflammatory and autoimmune diseases, produce interleukin 17, interferon gamma, and granulocyte monocyte colony stimulating factors, and express TBET and retinoic orphan receptor gamma T transcription factors. As regards systemic lupus erythematosus, pathway-based analyses have recently implicated several potential pathways, including thymic stromal lymphopoietin signaling in the immunopathogenesis of systemic lupus erythematosus. However, thymic stromal lymphopoietin expression was weak in specimens of discoid lupus erythematosus. As regards the role of thymic stromal lymphopoietin in alopecia areata, apart from the T helper 1 mediated immune responses in alopecia areata, analysis of the transcriptome. The transcriptome is the set of all RNA transcripts, including coding and non-coding in an individual or population of cells, revealed that an elevated inflammatory profile of T helper 2 genes, which include thymic stromal lymphopoietin, suggesting a potential involvement of T helper 2 signaling in patients with alopecia areata. A group of severe alopecia areata patients showed suppressed thymic stromal lymphopoietin related pathways and T helper 2 immune responses. In such cases, topical application of diphenyl cycloprobenone stimulated the thymic stromal lymphopoietin OX40 ligand interleukin 13 axis and facilitated the T helper 2 skewed immune responses, thereby restoring a balance between T helper 1 and T helper 2 ratio. Thymic stromal lymphopoietin was recognized for its capacity to link the innate immunity to the type 2 acquired immune response at the barrier surfaces. However, recent data demonstrate that thymic stromal lymphopoietin can also indirectly or directly affect various immune and non-immune cells involved in a diverse range of cutaneous immune-mediated disorders beyond atopic dermatitis, which significantly broaden the functions of thymic stromal lymphopoietin in different skin conditions. And in a retrospective study, a group analyzed samples from eosinophil-rich dermatoses found that thymic stromal lymphopoietin was broadly expressed in arthropod bites and drug rash, atopic dermatitis, bullous pemphigoid, and non-bullous eosinophilic spongiosis. These results, combined with the finding from the literature, indicate that thymic stromal lymphopoietin is strongly implicated in various skin conditions, not limited to allergy and autoimmune. Furthermore, thymic stromal lymphopoietin expression is elevated in patients with cutaneous T-cell lymphoma. In the presence of thymic stromal lymphopoietin, tumor cells expressing thymic stromal lymphopoietin receptors generate interleukin 13 and interleukin 4. In mouse models of skin cancer, a tumor suppressive effect for thymic stromal lymphopoietin was demonstrated by the keratinocyte-specific deletion of the Notch pathway. Furthermore, thymic stromal lymphopoietin has been linked to the pathogenesis of keloids. So we can conclude that the comprehensive understanding of the diverse properties of thymic stromal lymphopoietin in various immune pathways will be critical for future research and the development of effective therapies targeted towards thymic stromal lymphopoietin for the treatment of these conditions. I hope that you have enjoyed this somehow long video, and before we finish, I want to remind you again to subscribe to the channel and activating the alarming bell if you are interested in getting any new video, and thank you.